this is a final class, and then today we'll introduce the how to image or visualize actin, especially actin dynamics in live imaging. So, like this time, I'll briefly introduce actin and actin structure, and then I will introduce several methods or probe to visualize actin and the actin dynamics. So finally, we'll introduce this paper about how to visualize actin or the interaction between actin and the organelle in live imaging. Okay, so as you may know, actin is one of the cytoskeleton. It belongs to microfilament. And there are several functions, including they can support the cell structure, like they can locate in microvilli or the surround to the cell cortex or in the adherent belt. It can also help the cell moving, so it can localize the leading edge or lemipodium or filipodium, and you use myosin to help cells moving. So it's also involved in the endocytosis process. So when cells want to do the endocytosis, the actin mesh will like surround to the membrane and then help to do the invagination. They also can transport the vesicles, and then during the cell division, they can form the contract ring and help the cell division. So actin is formed from the actin monomer called G-actin. So in G-actin, this is a structure, and then you can see there in the center there are ATP binding cleft. So ATP or ADP can bind the bottom of the cleft, and they form from the four subdomain. So G-actin is pretty small, and it's about 5.5 nanometer. And then F-actin is the filament of G-actin. So G-actin forms F-actin, and then it's also very small. The width of F-actin is about 7 nanometer. You can see here the ATP binding cleft face to the minus N, and the other side face to the plus N. So actin is dynamics. It's not like form the F-actin and then stay there. So they can associate and dissociate at the same time. So when actin bind ATP, the ATP bound actin will bind the plus N of the actin filament. And the ADP bound actin will dissociate from the ATP filament. This also needs several actin binding proteins like profilin. So when profilin bind to actin, they can open the cleft. So ATP can come into the cleft to exchange the ADP. So profilin, ATP, and actin will come to the plus N of the F-actin and then bind to the plus N. So in addition, the profilin-bound actin monomer cannot bind to the minus N. And the dissociation of the actin ADP also, profilin will bind that and then help the dissociation. So this is the cycle of the filament turnover. So how actin filament polymerize? It requires a nucleating protein called formins. So formins, it forms a dimer, and they have several important domains. The first one is the Rho binding domain, can bind the Rho GTP protein, and the FH1 is the domain to recognize the profilin, and the FH2 is the domain for the nucleation. So after the RBD domain binds the Rho, and then the profilin with ATP and actin will bind to FH1 domain. Then FH1 domain can transfer the actin monomer to the FH2 and then form the actin filament. So in addition, actin is not always like a straight. It can form the branches and become like a mesh mesh work. So to do this, they require the other nucleating protein called the R2-3 complex. It also requires the nucleation promoting factor, NPF. So briefly, the actin can bind the NPF first, and then bind to the R2-3 complex. The whole complex will bind to the site of the actin filament. Then the actin on the NPF will transfer from NPF to the R2-3 complex with the minus end, and then it will form the branches. So the branches is about 70 degrees. So since the actin is very thin, so it's hard to image, and then it's very dynamic. So many groups try to develop the method or the probe to see the actin without affecting the actin dynamics. So now we'll introduce several different ways to visualize actin filament, especially in life. So the most straightforward method is, okay, then we just take or fuse GFP to the actin, or for other fluorescent protein to actin. But because the actin is about 40 kilodalton, and then GFP is 28, so it's pretty big. If you directly take the GFP to actin, it can affect the actin function and the dynamics. So that's why different groups develop different methods try to visualize actin. The first one, they use the small molecule like the well-known phalloidin. And the second method is they don't directly take GFP into actin. Instead, they take the GFP to actin binding protein. So like more indirect way. And the final method is the chromobody. So we'll talk about this. Okay. So the first method is use the small molecule. The very famous one is phalloidin. Probably most of you heard about that. So phalloidin is the derivative from the phallotoxin. It's one kind of toxin from the mushroom, and then it can bind F-actin with very high affinity. So because it can bind F-actin with high affinity, so after it binds to the F-actin, it indeed stabilizes the F-actin. So the actin dynamics will be affected. So it's not good for live imaging, but it's really good for the fixed samples. So basically, if you want to see the actin in your cells, you need to fix first, and then you use phalloidin. Then you can see the very beautiful actin filament in different cell types. It can use in live imaging, but then you can clearly see the actin dynamics is affected. You still can use live imaging to see the actin structure, but only for structure. You cannot do it already affects its function and the dynamics. So you use different image methods of confocal, wide field, even super resolution. You can clearly see the phalloidin staining on actin. So this is from Budding East. So this is wide field, this is super resolution. And this is the moving cells, this is the leading edge. This is from confocal. So anyway, for the phalloidin, it's great. It's the gold standard marker for actin filament, but only in fixed cells. For the live imaging, it's better to use another way. Okay. The second method is also from the small molecule called SIR-actin. It's also derived from another toxin, and it's from the marine sponge. So this one, because it's similar to phalloidin with different structure, it's also stabilized the F-actin dynamics. So it's also good for the fixed samples, but it's not that good for the live imaging. Although people apply this one to the live imaging, in certain cell type it seems fine, but then in most of time, you can see the morphology is change, and then the dynamics also change. So in the recent publication, they showed the structured data to explain how phalloidin or SIR actin affecting the actin filament dynamics. So as I mentioned, the ATP bound actin will bind the plus end of the actin filament, and the ADP bound actin will dissociate from the actin filament. So when ATP bound actin bind to the plus end, it starts to hydrolyze ATP from ATP to ADP plus the phosphate group. And one of the structure in the actin will change, so called the D loop. So in this is the D loop. So when they just bind, it will prefer the open form, and then they will gradually become a closed form. So after hydrolyzed ATP to ADP, it become that structure on actin become the closed form. The D loop in the actin become the closed form, and the closed form of actin will dissociate from the F filament. Sorry, and then the phalloidin and the SIR actin, after they bind to the ATP bound actin, they help to the ATP dissociate to ADP to PI and the phosphate group, but they cannot change from open structure to closed structure. They will maintain to the open structure. So the actin cannot dissociate from the filament because it's kept in the open structure. So that's the basic idea why phalloidin and SIR actin can stabilize the actin dynamics. They also have different effect on the F filament. For phalloidin, if they bind to the ADP bound actin, they will keep them in the closed form, so they cannot change back to the open form. But for the SIR actin, they will keep everything in the open form. So, but the key step to stabilize the F-actin dynamic is here. So when ATP hydrolyzes to ADP and the phosphate, they'll gradually from open form to closed form. But the phalloidin and the SIR actin will stop this process. They'll keep that always in the open form. So the F filament will stabilize. That's why on these two, you can clearly, when you use these two standing, you can see the very beautiful actin structure, but it affects dynamics a lot. Okay, so far it's okay? Okay. And then that's why people start to think, okay, we cannot direct fuse fluorescent protein to actin. It affects the function. And then for use the small molecule, it also affects the dynamics a lot. So people start to think, so we can fuse the fluorescence protein into the actin binding protein, so they won't affect the actin polymerization and the dynamics. The first one is from yeast. It's the ATP114 protein from yeast. So this one, BNR1, BNR1 is the forming in yeast. So this is the forming, and the F filament start to polymerize. And what is ABP140? ABP140 is the protein for the actin cable bundling. So in yeast, after the F filament form, they will bundle together, become like actin cable. So in yeast, so in most of cell type, the main track to moving cargoes are like microtubules, but in yeast is actin. So they will form the actin cables and become a track for like mitochondria moving, other vesicles moving. So ABP140 is one of the protein for the actin cable assembly. So we can take the fluorescent protein to the ABP140, and then they can perfectly show the actin filament without affecting their dynamics. So this is first found in 2002, and this is from my postdoc lab. So when my postdoc PI, she's a young PI, she developed this. So you can see this is ABP140 GFP, and then this is, you can see the actin filament, and you can clearly see the actin polymerase and from the bud neck to the bud end. And if you compare the stenophyloidin and the ABP140, you can see it's almost 100% overlap. There's one exception, is that in phyloidin you can clearly see this dot. This is called actin patches, which is the endocytosis vesicles. But then in ABP140, it's not that clear. So, but for the actin filament, it's almost 100% colocalization. So this is the first probe can visualize the actin filament and without affecting dynamics. Since ABP140 is found, different groups start to try to find a modification, try to modify the ABP140, and then find a good probe for actin filament in other cell type, because ABP140 only in yeast. So in 2008, they found that the first 17 amino acid of ABP140 is enough to label the actin. So if you compare the sequence of the first 17 amino acid of ABP140 in the this is button yeast Myces cerevisiae, the relative of this is pretty conserved, but it is absent from other organisms. So then they use the 17 amino acid of the ABP140 and then fuse with other fluorescence protein. Then in different kind of cell type, it's perfectly labeled the actin filament, and even in yeast, it's do the same thing. So from 2008 to like 2010, or like the couple years after this developed, it looks great. So in the almost cell type, it's because only for a couple of years, people start to test this, and most of the time it works perfectly. But then they found there's no any effect in the actin dynamics, and the actin looks pretty good. But then after several years, people start to find that in certain cell type, it also affects the morphology and the dynamics. For example, in the fly, Drosophila S2 cells, if you express the life act in S2 cell, the morphology of S2 cell can change it. And then for fission yeast, these are budding yeast. For fission yeast, if you use life act, it affects the cell division. So actually, it also affects some cells. But for the most of the time, it seems fine in using life act. So if you check several literature, life act is one of the most used method to visualize actin. So in very recent, so it's also published in the Nature method. They developed another method, the modified version of life act called LILAC. It's light-induced localization to actin. So it's the optogenetic version of life act. So the idea is very simple. So they just fuse the LOVE2 domain, which is the well-known domain used in optogenesis. It's from the phototrophin 1. So if you shine the 450 light to this LAV2. The original, the J alpha helix and the A alpha helix like fall together. After you shine the light 450, they'll open and then can bind to the actin. So they also test several things. For example, without excitation, lilac can like diffuse because it's like kind of monomer diffuse in cells. After you shine the light, the 450 excite, it go to the actin filament and label them. Then if you remove light, it will recover. So the good thing is, for the traditional live act, it can bind the F filament, but for this optogenetic one, you can control when do you want to see the filament. When you want to see the filament, you can shine the light, then you can see the filament. And normally, when they grow, the live act won't affect the actin dynamics. So after they remove the light, they can recover about after 1 minute they recover. So which means the lilac dissociate from the live actin filament. They also check the actin flow. So this is the chemograph. If you remember last time we talked about that, you can see this one. It's moving. And then you can do that repeatable. You can shine the light and then lilac bind to the F-actin, and then you remove the light, it will come back. And then you can repeat several times without affect the cell function. So this is a newer version of the live act. So other groups used a similar strategy to develop the probe to visualize actin. So for example, this F-tractin is another probe. So F-tractin is the modified version of the inositol triphosphate kinase A. So it's the 10 to 52 amino acid of ITPKA. And then they also found it's enough, this amino acid is enough to label the actin, and in some cell types it won't affect the dynamics. Similarly, eutrophin is another way. Eutrophin is the homologue of dystrophin. So endogenously, it's found in the fetal or regenerative muscle, and after that, the eutrophin will replaced by dystrophin. So I can use the eutrophin fused with fluorescence protein and then to see the actin dynamics. So basically, from phalloidin, F-tractin, and eutrophin, it's 3 different methods. So these are 3 different actin binding protein, and then they fuse with GFP to visualize actin filament. But most of people still use live act because it's only 17 amino acid. It's pretty short. It probably has less effect on actin dynamics compared to these 2. Okay, so the final one is chromobody. If you, in these 10-15 years, this one is developed. So what is chromobody or nanobody? It's for the conventional antibody, you may know it have the light chain and heavy chain. But in alpaca or llama, they have another kind of antibody, which only have heavy chain. So you can take the variable region of a heavy chain and then fuse with fluorescence protein. And then this one can recognize actin, or you can design this antibody to recognize different stuff. So you fuse with fluorescence protein, and then if this one is specific for actin, then you can visualize actin. So far, this one seems the best method to visualize actin. They have minimal effect of actin dynamics. Although because it's in this about 10 years, probably after 20 years, people start to find there are some defect. But as this is so far, this one seems the best one. They have minimal effect on actin function and the dynamics. So they can also apply in different cell types and in plants. So this is the tobacco leaves. So if you have the slice, you can see, but it's not that clear here. So if you treat the actin with electron cooling B or lead B or lead A, so these are drugs to dissociate the actin filament. So you use the actin chromobody to visualize actin, and you treat the lead B to dissociate actin. You can gradually see the F-actin filament dissociate. After you wash out the lead B, actin can repolymerize, become the actin filament. So this is the one of the example to see the actin structure and the dynamics use the chromobody. Okay, so far, any questions? So this is the, it's a pretty good table for you as a reference. Oh, okay. I forgot to cite reference. It's the same one like, it's a 2017 one. Sorry, I should put the reference on that. And so this is the comparison of the all different kind of probe of actin. And then can see you directly take the GFP, they show some of the defect, and then for the phalloidin and then SIR actin is a small molecule we just mentioned. And the live act, utrophin and F-tractin, it's the fuse the fluorescent protein to the actin binding protein, and you can label that. So far, I think the first three is the most used method. The first, for the fixed sample, you can, phalloidin is the gold standard. So everyone uses phalloidin. And for the live imaging, depends on your cell type and your model organism. You can choose live act or others, or this actin chromobody. So actin chromobody looks great, but then it's not always can apply to different cell type. For example, we try to use actin chromobody in budding yeast, but we never see any signals. So you have to try. But so far for these three, I think it's the most used method to see the actin filament. Okay. All right. So although we have different methods to see the actin filament, it looks good. But then it's still hard to see the interaction between actin and the organelles. So that's why these papers published, they try to develop a method to visualize the interaction between actin and the different organelles. The limitation of current methods, although you have good way to visualize actin filament, because in a cell, the mesh work of the actin, so the intensity and the background ratio is very high. The signal to background ratio is pretty high. So it's, when you see the image, it's kind of blurred. And then the interaction between actin and the organelle is transient. It's very dynamic, so probably you cannot see that. And the resolution probably is not that good. So they try to develop the method to see. So the idea is that when actin interacts with organelles, then you can see the clear signal. So they try to use this way. So they fuse the organelle membrane targeting sequence to the actin chromobodies. So if you fuse the membrane targeting sequence, they'll insert to the double membrane of your organelles. so they can diffuse freely. When actin comes in, the chromobody will recognize actin, so they will concentrate to the interactin region. So after actin comes in, there are more actin, and so there are more chromobody, so the signal will become stronger. So is that okay? So this is the idea of how they develop this method. Okay. So this is a good method to study the actin interaction between actin and the organelles. So they developed two versions. One is AC mito, which is mitochondria interacted actin, and the ER interacted actin, ACER. So for the AC mito, they fused the EAST-FIS1 membrane targeting sequence to the actin chromobody. For ER is cytochrome B5. So this figure just shows that they can, its work. It can localize to mitochondria or ER. But notice that, so in this panel is AC mito or ACER. This one is M-cherry, I think. The M-cherry, so it's labeled mitochondria or ER. So you can see AC mito indeed localized to mitochondria, but they are not diffuse everywhere. It's like concentrated in some area. This suggests that this area is the spot actin can interact with mitochondria. And similar like this is ACER. You can see it's not diffuse everywhere in ER. It's only some spots, some areas. And then if you compare this one to mitochondria, so this is a mitotracer to label mitochondria, you can see some overlap. So if you remember last week when we talked about the mitochondrial fission-fusion process, during the mitochondrial fission is require actin. So this part probably is like ER with the actin comes in and then help the mitochondrial fission. So they also shows this one is can do the time life imaging, and then for the A figure is the ACER, and they also use life act to label the actin and try to make sure the ACER is really labeled actin. So it's it's co-localized with live actin. And this panel is the AC mito. In different time points, you can see AC mito also localized with live act. But then, if you see that, for example, like this one, this is the AC mito. But then, if you use live act, you can see this very strong background because there are other actin in the background. So, but this one you can specifically shows the actin which it interact with mitochondria. Okay, so after they show their method is working, they try to use this one to study the dynamics of actin with mitochondria during the mitochondrial fission process. So if you remember mitochondrial fission process, last week I said, okay, ER comes in and then actin from the ER and then bind to the mitochondria, form the first contract, and then recruit the mitochondrial fission protein DRP1, then mitochondrial fission. But it's still not that clear, the temporal spatial is not that clear about the mitochondrial fission. So they can use this method to see the actin, how actin control the mitochondrial fission. So further, they find that AC mito, which means actin mitos, accumulate before the mitochondrial fission. So here is the mitochondria. This is a hollow mito, which is another way to label the mitochondria. So see the mitochondria before fission, you can see the AC mito signal increase, and then after fission, the signal decrease. So this means that AC mito accumulate before the mitochondrial fission, which is consistent with the other studies. Actin comes first and then help mitochondrial fission. Similarly, when they check the ACER, the ER-associated actin showed the same thing. So the left side is the AC mito, is the same idea like the previous slide. So this is a mito tracker rate to label mitochondria. This is the AC mito. So before mitochondrial fission, AC mito already accumulate, and then they will fission, and then the signal disappear. Similarly, in the ACER, they show the same thing. Before the mitochondrial fission, you already can see the ACER signal increase, and then after the fission, the signal decrease. So this all consistent with the previous studies that actin comes first and then help the mitochondrial fission. I think the interesting part is this one. They find that AC mito indeed accumulate before ACER. So this is mitochondria. Before mitochondrial fission, you can see the AC mito signal first, but you didn't see the ACER signal. And then if ACER signal increase, but you still can see the AC mito signal. And then mitochondrial fission, then two signals disappear. So you still don't know how to explain this. The AC mito accumulate before ACER. And then I think it's a great way to do for the further study. But they indeed show that this ACER or AC mito it's accumulated prior to DRP1, and this is also consistent with previous study. So you can see this is the ACER, and this is mitochondria. So this is DRP1, and then you see the ACER accumulate first. Then they DRP1 is recruit to the mitochondria, and the mitochondria fission. So this is also consistent with the previous study. And similarly, like the AC mito, it's the same thing. So you can see this is the mitochondria. This is a DRP1 signal. Here is the AC mito. So AC mito here accumulated first. This time there's no DRP1, and then DRP1 shows up, and there's AC mito still there. And then at this time, mitochondria still fused together. After this time, mitochondrial fission, and then signal disappear. So they use this method to just they proved several previous studies like actin comes first, then DRP1 comes in, and mitochondrial fission. But previously, the model is the INF2 is the forming from the actin filament polymerized from ER and then attached to the receptor on the mitochondria. So if in this case, we should see the ACER comes first and then AC mito comes later. But in here, they say they found that AC mito accumulate before ACER. So where the actin come from and how the mitochondria actin interact with ER, I think it's a new topic we can study further. Okay, so I think that's it for today. So any questions? No? Okay. Okay, then that's it. All right, thank you.